I'm Jim Heath. I'm the president of the ISB and professor here and also professor at UW. And I'm delighted to be presenting this research roundtable today. So I'm going to share screen as I continue my introductory remarks. As president of the ISB, I oftentimes get asked, do you guys work on cancer? Do you work on infectious disease? Do you work on chronic disease? Do you work on aging? Do you work on health? All these different aspects of the human condition. And we're a relatively small institute. We're located here in South Lake Union, and I always answer yes. And the reason why I answer yes is because the essence of the ISB is that we take a systems biology approach towards understanding the human health condition and variations of it. And so the talk I'm going to give today on using generative AI to begin unraveling secrets of the immune system is probably as illustrative of a talk. In fact, we will look across aging, we will look across cancer, infectious disease, healthy, etc. Everything from the fetal thymus all the way up to senior citizens, to try to look across diseases and put together general rules of how the immune system works using this novel and still relatively new tool of generative AI. So this is the essence of systems biology, and I'm delighted to be here to talk about it. If you want to learn more about the ISB, you want to participate, you want to donate, you want to have some role in what we do, please reach out to us at isbscience.org. So if you are a scientist or biologist going through college or grad school, you're likely to, especially if you're in biology and human biology, you're likely to take a class in molecular immunology. And the majority of what you learn from an immunology textbook is really based upon studies of lab mice. And that's because in lab mice, you can do all sorts of perturbations. You can look at things over a function of time, even on a daily or even on an hourly basis if that's necessary. And in many ways, mice have immune systems that somewhat mirror that of humans. But in many ways, they're wildly different. They're designed to live for 2 years. We're designed to live for on the order of 100. They eat a simple diet. They live in sterile conditions. And so a consequence of that is that the human immune system is far more diverse. And that richness is not just in each one of us, but it plays out in how we're all different from one another. And, at the heart of why people have different chronic diseases, or oftentimes different cancers, or age at different rates, is this diversity of the human immune system. And this is something that has fascinated me for quite some time, and I'm going to really embrace that today. So I'm going to give a very brief primer for the scientists who are on here. I apologize for this, but it's only going to be a few slides. So here's just in one snapshot your immune system. Most of your immune cells, in fact, all of them come out of the bone marrow, hematopoietic stem cells. They differentiate into two branches. One is called the innate immune system, which has cells like dendritic cells or neutrophils or platelets, things that you possibly have heard of if you have just had labs done in a clinic. And on the right side are the cells that have memory, but do quite a bit more than that. And I'm going to talk about T cells today. But B cells are another part of that component, and as are NK, or natural killer cells. So at first glance, there's two types of T cells. And I'm generalizing here. This is not strictly true, but at first glance. So there's CD4 T cells, which are also called helper T cells. And these are in many ways the conductors of the immune orchestra. And then there's CD8 T cells, which are called cytotoxic T cells. And as you might guess from their name, these cells kill cells that present non-self signatures. And cells that can present non-self signatures might be infected cells, virally infected, or cancer cells. And cancer cells would look not like self because of, for example, mutations. So to understand how, for example, a CD4 T cell is an orchestrator of the immune system response to, say, a pathogen, this following series of cartoons more or less illustrates this. So we have this up here in the top left, we have what's called a macrophage, part of your innate immune cell, that's innate immune system. It will actually eat a pathogen. It will physically eat it, called a process called phagocytosis. And a fragment of that pathogen can be displayed on the surface in, with what are called MHC molecules, but it's put on the surface so that T cells that are swimming around in your body or your lymph system, if they encounter this little fragment and they recognize that fragment as being foreign, then they will activate the T cell, and that T cell then will go to a B cell and say, perhaps look out for this. The B cell can mature and it will produce antibodies. And so this is a way in which your T cells, which are really designed to look at things that aren't you, non-self, can kickstart a pretty strong immune response against a pathogen that helps your body to remember that pathogen the next time you see it. A cytotoxic T cell, on the other hand, is very different. A cytotoxic T cell also has the ability to recognize things that are not self. But once it recognizes that, for example, a fragment of that non-self virus or something like that, that's presented on a infected cell or a tumor cell, it will just kill that cell. And in terms of cancer immunotherapy, most cancer immunotherapies are really designed, for example, to activate CD8 plus T cells. And this picture on the bottom right is of a melanoma tumor. That's an actual patient, a tumor that was resected. And what you're looking at here on the left side is T cells that are just invading and taking over and killing tumor cells. So T cells are killers if they're CD8, and they're immune orchestrators if they're CD4. The thing that makes T cells different from other immune cells is the T cell receptor. And so each of us have about 1.8 trillion, a big number, 2 times 10 to the 12th. And the vast majority of those nearly 2 trillion T cells will have a unique T cell receptor. That means that it's one protein, but there's a piece of that protein called the hypervariable region right here that actually gets shuffled from T cell to T cell. And that's what gives our immune system the diversity to recognize virtually any foreign entity that gets into your body. There's some other fun facts. How much do the T cells in your body weigh, et cetera. But a question that I think many people would have when they hear about this is, how are the T cells so programmed to see things that are not self? Why don't they have aberrant reactions with self? And occasionally they do, and that is an aspect of chronic disease. But the reason why in general they don't is because of a process called thymic selection. So I told you that your immune system comes out of your bone marrow from hematopoietic stem cells. T cells travel to the thymus, where they mature, and they're exposed to pretty much everything they're likely to see that is self. That's the goal of the thymus, is to expose T cells to all the kind of things that look like you. And if a T cell gets activated in the thymus because it saw a piece of you, then that T cell is killed. And so only the T cells that are not self-reactive, and that's really like 1 in 10 or 1 to 2 in 10, will make it out of the thymus. And so it's this selection process that is essential for T cells to recognize foreign antigens, whether they're helper T cells or cytotoxic T cells. And it actually gives you a little bit of a clue on aging, because as you go from a fetus to a newborn to a little kid, all the way up through adulthood to senior citizenry, I don't know if that's a word, but senior citizenry, your thymus will lose function. It is at its maximum as a fetal thymus or a newborn. and it's producing the T cells that you're going to have mostly for your life. It'll continue producing new T cells for a certain amount of time, but by the time you're, middle-aged, your T cell is basically just a glob of fat. It's no longer functional, and so you're not going to generate any more T cells to first order. Maybe very few, but by and large, the T cells you have by the time you're at middle age are the T cells you're going to get. And so when you look at aging-related diseases, this is a big reason why, because your immune system has not continued to evolve to, for example, challenges that you encounter, especially new challenges. Okay, so finally we get to generative AI. So what I've shown here on the left, this looks like just a bunch of random numbers that all start with CA. These are the amino acid sequences of the hypervariable region of a T cell receptor. These could be CD4 T cells, helper T cells, or they could be cytotoxic T cells. A trick here that we use is that in many ways, these look like words. So even though I know that you can see some similarities, like they sort of start and end in the same way, which is the nature of that protein, but in the middle, they have a tremendous diversity. And this is when I say your T cells are all different to first order, is because they have different sequences here in the middle. But these look like words. And what that means is that we can take some of the tools that are basically useful in ChatGPT and other AI algorithms, and we can use them here, but in unusual ways. And further, when we are using T cells for the AI applications I'll talk about, generally the T cell is receptor, this one hypervariable region, we can connect it to a vast amount of additional data. And that's something that the ISB is really good at. So we can say for this particular T cell that had this particular hypervariable region on the T cell receptor beta chain, we know who it came from. We know when it was collected. We know the age and the section of the genome of that patient. We know their electronic health record. We know their diet. We might know their microbiome. We know the proteins that are in their blood, the metabolites. We know their clinical labs. We know what other T cells and what other immune cells were present. And for the T cell that T cell receptor came from, we might know the expression of 20,000 genes just for that one T cell, the levels of 200 proteins, the rest of the TCR, T cell receptor gene sequence, et cetera. All of this extra data is what's called metadata. And so as we develop this AI algorithm to understand T cell receptors, this metadata is a major way to interpret that data. And I'll go through some examples of that. Okay, so what we did to develop this algorithm, and the first thing I did was I hired this guy, Daniel Chen, in my group. So just a few words about Daniel. He's just finished up a Marshall Fellowship at Cambridge, and he's about to start graduate school, an MD PhD program at UCLA. He began working in my lab, I think, when he was 14 or something like that. He was an undergraduate at UW, and one of the most remarkably productive people I've ever had in my lab, and he's just starting graduate school. And this is his the brainchild of Daniel, by and large. He and I spent the last couple of years trying to develop this type of a computational way to look at T cell receptors that would allow us to look at new biology. Okay, so we basically scraped the internet for data that had T cell receptors and also had this type of metadata that you see here on the right. And so these are the data sets. I just want to be complete. Much of this data comes from our own experiments, but much of it comes from other people as well. But what this data allowed us to do is to have roughly 1 million T cell receptors upon which to build this algorithm. And we called the algorithm Tarpon. I don't know why. I know the T was initially T cell and the A was initially antigen, but I forgot the rest. And this algorithm evolved over a couple of years, so we just call it Tarpon. Okay, so the way we start this, this is a pretty complicated looking slide, but we encode the TCR not with just the word CASSPDIEAFF. In this case, as you can see, that's the T cell receptor hypervariable region of the beta chain. But we encode it in terms of its amino acids. It's the physical chemical properties of those amino acids, which turns out to be the most important part, and then some genetic similarities between these amino acids. And so when you do something like this, now you encode the T cell receptor in these various matrices, and then this is basically the neural net architecture that is the heart of most AI type approaches. And what was found half a dozen years ago, four years ago or so, was that this architecture was really great. You just had to feed it a lot of data. And as you fed it more data and more data and more data, it actually began to evolve into things you could have conversations with. We're not quite doing that. We have 1 million TCRs on the scale of the numbers of TCRs, even in a person. That's not many, but it's enough to do what we want to do. And so you take these representations of the TCRs, you reduce them down into what's called a latent, which is a 42-dimensional representation of this whole million set of TCRs. And then if you feed a TCR into here, reduce it to its latent representation, then you can regenerate that TCR, okay? And this is how you optimize this latent. You try to feed in a million of these guys, you regenerate them on this side, and the better you do a job at regenerating this, the better the latent is. So that's the optimization of the latent. Okay, so one way that you do that is that you can take, for example, here's 2 different TCRs. You can see that they're actually very different in terms of length, in terms of sequence. We put them in, we take them out, and we do this many, many, many TCRs. And in general, Tarpon will regenerate the original TCR to about 0.1 amino acid in length. So basically it gets the length exactly right. And it gets the sequence to within typically 1 mutation, 0, 1, or 2 mutations. I think 1 is the most common thing. So what that means is that now we have a mathematical construct that seems to generally work for all T cell receptors. But there's a cool thing. This is what's called a convolutional variational encoder, autoencoder. And so here I have this sentence here. The trick of a CVA is that each of these TCRs is actually transformed into a probability distribution. And so when we're asking this algorithm, what TCR did we put in, it's responding by saying this is the most likely probable TCR that was represented on this latent, okay? So now I can say put in 2 TCRs that are very different, and I can ask the CVA to produce a TCR that has 15% of TCR1 and 85% character of TCR2. And in that way, I can generate new TCRs, and that's what makes this generative AI. And you might be familiar with this kind of approach. Here's an example of generative AI being used. You've got 4 faces. Who knows which ones are real and which ones are synthetic? But you have gradual transformations from one face to another using these types of convolutional variational autoencoders. That's the same trick that we use here. Pretty remarkable piece of software technology. Okay, so now let's use this tool and see what it does. So this is I think everybody remembers that we had an epidemic a couple of years ago. This is the coronavirus. The spike protein, everybody probably remembers that. That was the unique thing that sticks out here that basically allows the coronavirus to infect your cells. There's a little fragment of this receptor binding domain, the green part of the spike protein that has the amino acid sequence YLQPRTFLL. It turns out that is an antigen that is what's called highly immunogenic. And that means that you generate T cell responses. Many people will regenerate T cell responses against that antigen, okay? So how do we use that? So if we look here on the left, this is our 1 million TCRs from many people, many disease conditions, many ages, and I've just presented them in a map. It's called a UMAP. It's just a way in which we can take 1 million highly diverse things and reduce them into a two-dimensional view. And even though you can't tell in this picture, this is like a Seurat painting. In other words, every T cell is represented as a dot. There's just 1 million of them, so it looks like a continuous flow. But each of these dots then is as a unique T cell. And if you're really close together on this map, that means that you're a similar T cell. You have similar chemical, physical characteristics. If you're far apart, you're very different. And so if we look at all the T cells that are reported to bind to this particular SARS-CoV-2 spike antigen, they all sit in one part of this representation. So we can fit that representation, even though it's, it seems to be spread out. And in fact, it's actually a 40 some odd dimensional representation reduced down to this 2 dimensions. We can actually fit it to something called a Gaussian distribution, and using an algorithm called normalizing flows. And then we take that distribution and we ask Tarpon, can you generate new TCRs that fit that same distribution? And it does a great job. And now we can even do a couple of pretty cool things. We can take T cell receptors that we know we see in patients from this experimental data set, and they've been validated. We can leave them out of the training. And then when we ask Tarpon to generate T cells against this antigen, it will actually regenerate these never before seen TCRs that we know are real. So in this small, but I would argue very non-trivial way, Tarpon can generate a T cell immune response to an infection. That's pretty awesome. That's the kind of thing that I think we've never been able to think about doing before. Okay, so let's take another example. So this is the fetal thymus. This is the place where your whole T cell biology starts. As I said, you've got T cells that come out of your bone marrow, out of your hematopoietic stem cell system. They travel to the thymus. In the thymus, they're what's called either double negative or double positive initially, meaning they haven't decided whether they're going to be helper T cells or CD4s or cytotoxic T cells or CD8. But at some point, they'll evolve into a CD4 T cell or a CD8 T cell. Okay, so using this particular data set that was published just a couple of years ago on 25 embryos and fetuses that were 4 to 17 weeks post-conception, that for whatever reason were abandoned by the body. It was basically miscarriages. We were able to look at these fetal thymuses and the T cells in them, and what you see is that with Tarpon, this algorithm, we can actually resolve the entire developmental biology of T cells in the fetal thymus. We can resolve these double negative and double positive T cell receptor just using the hypervariable region of the T cell receptor. That's all. Each of these dots represents a couple thousand T cells. It represents one of these fetal thymuses, and so we easily resolve the CD8s from the CD4s. And here's just a density map. So, so this says that, and this was not appreciated, you could do this before, that just based on those hypervariable regions, you can absolutely see what T cells are going to become helper T cells and which ones are going to become cytotoxic. To first order, those T cell receptors all look very, very similar, but not when you have this level of data. And in fact, that separation is not just good, it is almost perfect. And so it turns out that there's 2 of the dimensions in that latent, that thing that we train within Tarpon that most discriminate between CD4s and CD8s. If we take this discriminator and we look at 100 T cells that we would predict to be CD8s and 100 to be CD4s, and we ask, are they actually CD8s or CD4s? We don't miss a single one. That's remarkable. You never see that kind of thing in biology. And what that kind of suggests is that this environment of the fetal thymus has rules, and those rules are extremely well followed. And we can take those same rule sets and apply them to adults, and we find they actually work pretty well in adults too. So here's patients that have different, this is basically what we call a pan-cancer data set. So it's many patients with many different cancers, and we can readily distinguish the difference between a CD8 cytotoxic T cell, and a CD4 memory T cell. Here's T cells from patients with COVID-19. And once again, we can also tell the difference between the helper T cells and the cytotoxic T cells. And the discriminators in this case are pretty similar. Okay, so if we ask, this is just to show you that this is not a black box, what is it that makes the T cells different from each other, the CD8s and the CD4s, just by looking at that little hypervariable region of the T cell receptor? It's these chemical characteristics. So you can look at these characteristics, especially if you're a chemist or a biochemist or something, and you can say, well, I actually understand these terms. Hydrophobic means that it doesn't like water. Volume means that they're bulky molecules, forms hydrogen bonds. But the combination of these patterns is what's complicated. And that's the kind of thing that you can't pick out by eye. And so that's what these types of AI algorithms are very, very good at doing. And so if you have this combination of properties relative to this combination of properties, you're always going to be a CD4, you're always going to be a CD8. Okay, so now there's something really cool we can do. We have this pretty well-behaved fetal thymus. And so when we have all these other cohorts, healthy cohorts, cancer cohorts, infectious disease cohorts, so now you can do what's called a Bayesian-like question. So many people remember back in the early days when Obamacare was being formed, that there was a suggestion that fewer mammograms be incorporated into Obamacare. And it turns out there was a great uproar about that. But that suggestion was actually very scientifically sound. And it was based on asking the same question in two directions. So one question, if I have breast cancer, will my mammogram see a lesion? The answer is yes. The second question is, if I have a lesion that I see by mammography, does that mean I have breast cancer? The answer is almost always no. And because so many more people have no breast cancer relative to patients who have breast cancer, the mammogram has pretty low accuracy because of its false error rate, and it leads to a lot of extra procedures. In any case, the issue is when you ask the question in both directions, you don't necessarily expect to get the same answer, and not getting the same answer reveals new insights into biology. So if we take a fetal cohort and we train what's the difference between a CD8 and a T cell, and then we use that to predict on an infectious disease cohort, is not quite the same as training on an infectious disease cohort and predicting the fetal cohort, okay? And we can do all permutations of this for cancer, infectious disease. And if we had enough data, I would do this for aging and young and everything, but we don't have enough data yet. And what you find is that no matter how you train the data, your predictions on the fetal cohort are almost always nearly perfect. And so if I have, this is how good the prediction is. If it's 1, it's perfect. If it's 0.5, it's a coin toss. And you can see we always do okay. You know, we're at 0.85 or better almost always. But for the fetal thymus, we're almost always near 1. And in fact, if we train on the fetal thymus and predict on the fetal thymus, we really don't make a mistake, okay? So what that suggests is that the fetal thymus provides that rule set that I talked about, but it also suggests that in these adult cohorts, those rule sets can break down, okay? And those are rules violations. So a very interesting thing you can do is actually ask, well, what are the T cells that violate those rules? And what does that mean for those patients? And so here I've plotted the patients with COVID-19. And what you can see here is that here's the cytotoxic T cells, here's the helper T cells. The helper T cells mostly stay in their lane. They're mostly over here. The cytotoxic T cells actually stretch out quite a bit into the helper T cell area. And what that means is that you've got CD4-like CD8 T cells or helper-like cytotoxic T cells. And it turns out most people don't have those, but you see them in older men more often than older women. Something like 20 individuals out of 300 adults that we had. And those older men do much worse when faced with COVID-19. They had much more severe disease. I mean, you can look at this. This is on a scale of 1 to 7. For 7 means you're probably not going to survive, and 1 means you don't even know you've been infected. And so if you have really strongly biased CD8-like CD8 T cells, you really never get beyond a mild or moderate infection. And you do definitely head into serious infection land if you don't have those. And if you look in the blood of those patients, you saw that inflammation got out of control. And if you actually look at the T cells themselves, we have all the single cell data on these T cells. They simply do not act like CD8 T cells. They have no capacity to kill. And so these patients that had these rules violations, what we're finding is that they simply did not have the capacity, that initial front line of defense of CD8 T cells that identify and kill the infected cells. And it had health consequences for them. And so I'll conclude here and just tell you some of the lessons that I think we've learned here. So this application of Gen AI to T cell biology was a first. It's like, in many ways, I mean, I just touched on a handful of the observations that we made, but it's like putting on a new set of glasses to view the immune system. It's incredibly revealing. Some early lessons. You know, it allows us to quantitatively look across age and health and disease and genetic history, because we can make all of these Bayesian-like comparisons, and we actually have one set of human T cells, especially these fetal thymus and in little kids, where rules seem to be extremely well followed. And so we can look for rules violations. We can actually generate a T cell immune response against a pathogen. We have to learn about that pathogen first, but right now we can do that, and a never before seen immune response, but one that could work, because we know we can actually generate ones that have been seen before, just not trained on. Tarpon resolves T cell maturation in the fetal thymus, and it resolves those rule sets. And those rule sets are violated in some adults, and there are health consequences. I just talked about those health consequences for COVID, because that's right now where we have the deepest data set. But this almost surely provides a novel and quantitative view into chronic disease, cancer, aging, and things like that, that I think is it's like putting on a new set of glasses again. And so let's just go through some of the numbers. So we did all of this with 1 million T cells. That's only 1/1,000,000th of what an individual has in their body. In a couple of years, we hope to have data sets with 100 million. And so if you go from 1 to 100, you would expect to get the square root of new information. So that's 10-fold times more information that we would expect to get, okay? And that's going to allow us to look into these issues of how these variations in human immune diversity impact things like cancer and responses and chronic to therapies and chronic disease and aging, et cetera. We will almost surely see connections between human diversity, human immune diversity, and environmental factors and things like this. I believe we will be able to anticipate which patients, cancer patients, this is something that's very near and dear to my heart, respond to which immunotherapies, and which patients will need help to respond, maybe additional treatments. And I think, you know, we're just beginning to unveil the fact that there actually do seem to be design parameters that are well followed. And that's kind of adding a flavor of physics to biology. Biology has never really had rule sets before. It's had observations. And so to be able to identify some of these rule sets, I think, is a really big deal in terms of having a fundamental grounding in how to think about the human health condition. And with that, I think I am done. And I'm happy to answer any questions. I think we probably have some in the chat. Okay. Okay. Okay, one question is, are you using a mean and standard deviation? Did you actually test the data was a normal distribution? If not normal distribution, using these results in unsafe conclusions, then I guess can result in unsafe conclusions. That's very antigen dependent. So it is something we've explored. In particular, on this one epitope, the antigen, the YLQ epitope, which we developed a T cell response synthetically that pulled out public known TCRs. There was a recent bioRxiv paper put up by Tom Shoemaker in which they took 6,000 of these T cell receptors that had been reported to be specific to YLQ. They tested every single one. They cloned 6,000 T cells, and they found that about half of them were specific and half of them weren't. We got that hold of that data set, and we were able to resolve which ones were specific and which ones were not. And the ones that were specific, we were able to resolve two different binding motifs of the antigen to the MHC complex that presents it. And in fact, that's consistent with literature. I just learned that yesterday. Those two distributions are two different binding motifs do follow Gaussian-like distributions. Overall, it forms what you call mostly a Gaussian distribution, even with the ones that aren't good fits, because that's an inhomogeneously broadened distribution. And so just the thermal noise or fluctuations or what have you goes into that distribution would wash out small deviations. But of course, it gets better resolved when you know the antigen, you know the T cell, and you know it really, really works. And so we do know that. Many of these data sets that are public out there are also fraught with errors. We've done a fairly good job of trying to make sure the ones that we report are not, but you can't always clone every single T cell to know. But it's a fine question. And so if you generate a T cell that you think is interesting against an antigen, to your point, for sure, you don't want to use that unless you do really exhaustive testing on that. And this is something that we actually do a lot of in my lab. So I'm pretty aware of that. Okay. What are the implications of rule violations in newborns and pediatric cancers like neuroblastoma? It's a great question. So, pediatric cancers are very, very different from adult cancers in that they typically are dominated by some major highly penetrant defect, genetic defect, and how that impacts the immune system or not, I don't know. This data set which came out of the UK did not have such aberrant thymuses, at least according to the genetic analysis because the whole genome was collected as well. So for all intents and purposes, they look like healthy functioning thymuses. I think it might be a very different case if you had a childhood defect that was highly penetrant and leading to something else and leading to some disease condition. And so I think that's a whole different area for study. What are the drivers in this rule breaking that's occurring in adults? That's a pretty interesting question. It could be that as you get older, there's many things that could happen. So the fact that we see this in predominantly men in this infectious case, and I'm sure we'll see this in other cases as well, but this is the one where we had the deepest data. There is actually a loss of chromosome 13 in older men. And that's a really tiny chromosome. It only has a handful of genes, but it's known to affect the hematopoietic system. And so, in fact, its major impact is on the hematopoietic system. And so there could be an interplay there that we don't resolve. It could have to do with the fact that the older you get, even though you can still produce some T cells from your thymus, the less effective that thymus is as it begins to shut down. So, so I think that those are our best guesses right now on why these rule sets break down in adults. I think we need to have quite a bit more data before we have a deeper understanding of what that means more broadly. How large are the data sets used to train your models? We used only 1 million TCRs to train these models, but we did a pretty careful job of curating those TCRs ahead of time. We obviously didn't clone every single one, but we tried to take them from what we considered to be the highest quality databases out there. It seems like, okay, here's a question. I think I've kind of answered that one. Here's a question. I like the first part of it. Amazing talk. Thank you. I have some experience on training a variational autoencoder model locally. My question is specifically on the computational resources usage on current. Okay, this was we used an old GPU system, and it takes about 1 minute to resolve this. So that was really the advantage of doing this with just the variable regions of these TCRs. We would probably get a better latent if we use the variable regions, and then there's other regions of the TCRs that are sort of semi-variable, that if we use the full TCR gene sets, and that's something that we're gearing up to do now. But at the moment, we just were able to get by with just these short words and generate all these observations. And it turns out, here's something remarkable. So the hypervariable regions are what are called VDJ. And V and J, so you noticed if you looked at these sequences that I showed, there was like CA at the beginning of one and F on the end. That's the V and the J part. Okay, so those are not nearly as variable as the D part in the middle. If we lopped off the V and the J part and just used the D part, it still did a pretty good job. So most of the information there is just in the D part. That's a fundamentally new observation. It was known if you just took the V and the J and some of the other parts, because that's what sees what are called MHC alleles, that you could actually do some discrimination between CD8s and CD4s that way. But just looking at the central, the D part was new. How can you extend the longevity of the thymus gland? I have no idea. I can't answer that question. I think it's a good question, but I don't know the answer. If you let me see if I have other questions. Do I have other questions? Q&A. At the beginning of your talk, you mentioned students learning about the immune system was based on lab mice. Given the power of generative AI and what it can tell us about the human immune system, it seems that we will have a totally new understanding. What does that mean for every immunologist with classical training? That's a really good question. I'm not an immunologist. I don't have classical immunology training. And, the modern approaches, there's two things that have really happened in biology that I think have dramatically changed how we think about immunology. The first is the ability to resolve the confounding heterogeneity of biology through single cell methods. And that's been a revolution that has happened really over the past decade, to the extent that if you're publishing a paper in a high quality immunology journal and you're not doing highly multiplex single cell analysis, you're probably not going to get it published. And so that has had an amazing resolving power. But what it's meant is that people like us who do this as a matter of course, and you can also look in the blood and measure 6,000 proteins, et cetera, are just swimming in data. And you've got the genome, et cetera. And so are there methods that we can use to look across all these data sets and actually reveal what I called immune rules or whatever? I think this is an example of such an approach. I believe as these data sets and as these methodologies become more and more integrated, I think it will shed remarkably new light on the diversity of human immunity. And I would say if you're a classically trained immunologist, I wouldn't forget everything you learned, but I think it's going to be a real opportunity. That's probably the right word. Opportunity to learn new immunology as these tools come along. Okay, you are doing this cutting edge work when generative AI is in its infancy. How do you imagine research in healthcare will evolve in the next couple of years as technology advances? So I think there's two sides to how generative AI is being used in healthcare. I'm talking about using it at a level of very fundamental biology to try to understand. I like this concept of fundamental rule sets and deviations from them, and that's a hypothesis that we will pursue over the next several years. There are many other approaches that are basically taking electronic health records and physician decision processes and et cetera, and trying to have either AI physician assistants or even more or just AI deciders on trying to do disease diagnostics, trying to look in unstructured data sets like MRIs or CT scans or H&E scans of resected tissues. There is an incredibly strong financial motivation behind all of that because it actually translates directly into the efficiency of our healthcare system, and it makes physicians much better physicians if these tools are used wisely and they're developed and everything. That's a place where fundamental scientists like ourselves are probably not going to play that game, and using these tools at the more fundamental level to resolve biology is the other aspect. And so I think, you're right, generative AI is at its infancy, but it's not remarkably different from the sort of neural net stuff that we've done for a long time. This one thing I talked about where you use probability distributions as your mode of training and prediction is actually much newer, and it's just turned into a very exciting tool for us, but I think these algorithms, they continue to evolve. We'll continue to explore their value for fundamental biology. What causes T cells to misbehave in older men? I think I answered that already. What are the drivers of this rule breaking? I think I answered that. Do the rules you uncover allow you to distinguish between CD4, allowing you to distinguish CD4 and CD8, reflect the fact that CD4 and recognize MHC2, which are processed differently? Probably. So the question is, you have CD4 T cells recognize class 2 MHC restricted antigens, CD8 recognize class 1 restricted antigens. Those antigens are different, and they're differently processed. So is that what we're resolving here? Well, the fact that you can kind of see MHC versus 1 versus 2 by just the V and the J segments tells you that those two things are already predictive of, to some extent, of CD4 versus CD8. I don't really understand why just using the D segment works so well. And so I think that's something that we are going to have to explore quite a bit. That was, when we cut off the V and the J segments and our predictions remained almost as good as they were before, I got to say we were a little surprised. And so that's a result that, while a very true result, I don't think we fully understand it yet. Does it follow that CD4 and CD8 cells represent? Yes, that's absolutely true. The CD4 and CD8 cells recognize very different sets of antigens. How can we determine to be sure the functionality of the generated TCR in body system in vivo? So the way if you generate a TCR, you've got to clone it and test it. And then one of the reasons why we left out known TCRs is because those have already been cloned and tested. And so this shows that we could actually generate known TCRs, even with training on as few as 100 TCRs to give an antigen, which is a very small training set. If you actually wanted to use a TCR that you've discovered this way that is new, and you wanted to use it clinically, it has to jump through a lot of hoops. It's called an engineered T-cell receptor. And the FDA has established all kinds of rule sets that you have to jump through to get that, something like that allowed in the clinic. We do T-cell development for therapy in my group, and we, by and large, use just T-cells that we've identified out of patients who have the disease that we're trying to treat, so we know that they're safe in humans. But the ability to generate T-cells just opens up whole new opportunities that we for basic science. So we haven't really thought as deeply about as we could. But that's for next year. Do I have other questions? Have I answered them all? More in chat. Have you looked at the microbiome associated, in particular the metabolites of the microbiome? It's a great question. Sean Gibbons at ISB has done that kind of thing, and we have talked about trying to converge the types of approaches that we're doing here with the microbiome type of approaches, and I think that's going to be incredibly revealing. We haven't been able to do that yet, but I'm looking forward to it. I have some okay, I answered that question. Do you plan to continue to use the fetal thymus data as a baseline to determine rule-breaking activity, or take a different approach? It's a good question, and I don't know the answer to that. As we resolve larger and larger sets of, as we sort of build to, say, 100 million T cells that we can train on, or maybe 1 billion T cells, you may be able to resolve those rule sets through these Bayesian-like comparisons without actually using fetal thymic data, because deviations from the rule sets may actually begin standing out in ways that we don't really anticipate yet. So I think right now, because we have a relatively limited data set, it's been an incredible resource for resolving these rule sets. But in the future, I'm guessing that probably we don't need it, but I don't know. I think that's a good question. I think I'm done. Okay, well, and we're at the top of the hour. So thank you all very much for listening in, and come visit the ISB sometime, participate, do science with us, learn more. We do these research roundtables roughly 3 or 4 times a year, and we'll make a public announcement the next one. I think we probably have a hiatus for the summer, but we'll come back in September with the next one. So thank you.